Hello, and a very warm welcome to everyone joining us for this Select Science webinar titled Combining DNA Encoded Libraries with Machine Learning to Accelerate Drug Discovery. My name is Cameron Smith. I'll be moderating today's presentation, and I'm delighted to be joined by our guest speaker, Joe Franklin. Joe Franklin is the Senior Vice President of Early Discovery for Anagenex, whose mission is to accelerate the discovery process for new medicines by combining massively parallel biochemistry with machine learning. Please feel free to submit any questions you may have during this webinar. To do this, simply hover over the purple speech bubble icon on your screen and click to open the submission tool. Throughout the presentation, we will also be posting some poll questions relevant to today's discussion on your screen. If you could take a moment to submit your answer when prompted, it would be greatly appreciated. So without further delay, I'm delighted to hand over to Joe and would like to thank him for presenting to us today.
I'd like to thank the organizers for having me here today. I'd like to thank my colleagues for all of the hard work and great work that they've done to allow me to give this presentation today. And I'd like to thank all of you for your attention and for the robust discussion that we'll have after this presentation concludes. My presentation outline today will be to give you a brief overview of Anagenex, tell you a little bit about our DELs, finish up with some case studies that show how our technology platform works, and then that will lead us to some robust discussion following this talk. So what is Anagenex? Anagenex is a small molecule drug discovery company. It is our mission to discover, clinically validate, and commercialize new therapeutics for really challenging disease areas. How we will do this is by combining ultra-large screening techniques, such as DNA-encoded libraries, with machine learning. Our platform will enable us to look at more compounds more efficiently than ever before. We control our process end-to-end and have built it from the ground up. Our platform allows us to advance compounds more quickly to challenging targets. And our platform leverages and optimizes these molecules using machine learning. There are two main factors that limit small molecule drug hunters. The first is that all the easy targets already have drugs discovered for them. So what the drug hunter is left with is really challenging, really difficult targets. The second problem is that hit-to-lead optimization is too slow and too expensive. At Anagenex, we are looking to improve on these two processes. So as we're all aware, in the normal hit ID discovery process, you do some sort of screen, you then look at the hits that come from that screen, and you pick a few of those to move forward. You then start an iterative process of trying to make these hits better. And this process usually looks at one or two hypotheses at a time, and will make 5 to 10 compounds at a time. And then once you're done with that and you test the compounds, you come up with a new hypothesis to make your current best molecule better. You keep doing this iterative process of redesigning hypotheses, making a few molecules, and inventing a new hypothesis until ultimately you end up with a molecule that is ready for the clinic. Now, we all know that this is really difficult and really challenging and takes many, many years if you're lucky, because there is over a 99% failure rate of going from hit ID to patient. At Anagenex, we're looking to drastically improve the rate at which we go from hit ID to patient. We're looking to get here faster and with more options. How we're going to do this is by screening a lot of compounds under a lot of conditions, and not just evaluating a few hits to bring forward, but evaluating all the hits to bring forward. We will then use this data to empower machine learning models, which will then design a very large compound set to retest under our hypotheses. We will then use the data that comes from this second screen to empower a third screen, which ultimately is going to help us get here to the patient quicker and with more options. So to build our process, everyone needs to understand that machine learning will only be as good as the data you give it. If you give machine learning great data, it will do great things. If you give machine learning horrible data, it's not going to do very much for you. So with every part of our process, we have designed it to produce the types of high quality data that machine learning models need. In addition to producing that data, we've had it collated in a way that is easy for machine learning to use. So at the end, after this data goes through our proprietary models, we are able to predict highly accurate molecules that work against our target and design new experiments of large compound sets that help us get to the patient quicker. So how we do this is we do a DEL screen under many conditions against our diversity libraries. The output of this screen is used in a machine learning model, and this model does two things. The model will predict, based off of this screening data, commercially available compounds that look nothing like the screening set that should be hits against your target. So predicting out of library hits against your target. The model also will design a new library that can be screened and help inform the model better. And we can also ask all kinds of different questions from this library. We could look at selectivity, we could look specifically at certain phys-chem properties or ADME tox, but what we can do is use these two predict and design steps to then do another round of machine learning based off the data from this and predict a new set of compounds to test. At the same time, we can also use this data to predict a new library. And by repeating cycles of this, what we do is stay in high data mode as long as possible. We analyze more compounds, thus giving us better alternatives after each step. And we can make the questions we ask the model increasingly more difficult to bring in things like properties and other metrics, ultimately getting us here to the patient quicker. So what does this process of iteration actually get you? In yellow, I'm showing a UMAP plot of our diversity libraries. So this yellow in this screen is the chemical space of our diversity DELs that we screen on that very initial screen. This data is used in our model to predict the red, which are the commercially available compounds that the model predicts to be hits. As you can see, these red dots are in completely different chemical space from the yellow. The model also designs this maroon library, which is 175,000 compounds, different from the initial screen, that can be used in a rescreen to help the model better predict a second generation of compounds in green. So we start with the yellow, we end up with the green, we produce high quality hits that look nothing like our original molecules and are optimized for the properties that we desire for our program. So how is Anagenex different than the normal DEL machine learning company? We have a lot of experience. Our team has been doing DEL for 20 years, more than pretty much anybody in the industry. And our team knows what's really good about DEL and where the failures of DEL are. And we've designed each part of our process to make what's good about DEL the foundation and to develop on the failures. So make the failures go away. And so our chemistry platform focuses on winning DEL designs and not on having redundant molecules. Our selections process has been completely revamped, and the goal of our selections is to provide the clearest picture of how our molecules are engaging our target. Our analysis platform builds off of the data from these two and allows us to accurately predict hits and misses. So we're not following up compounds that don't show activity. And more importantly, we're providing machine learning with data that is accurate on compounds that engage our target and accurate on compounds that do not, and allows our machine learning process to make highly accurate predictions because the machine can tell what the hits and what the misses are. And as I've already mentioned, we follow up a lot more compounds than most people do. And so this is what our libraries look like. In a short amount of time, we've built over 30 libraries focused in these three types of areas. Our libraries are well balanced between 2 and 3 cycle designs, and our molecules are very drug-like. If you want to use machine learning to predict drugs, you should show machine learning compounds that look like drugs. One of the keys to us being successful here at Anagenex is that we're able to make libraries quickly. Our evolved process where we make iterative libraries depends on being able to turn libraries around quickly. If we were slow, our process would be too inefficient and we wouldn't get to the clinic faster. So how do we do that? We've completely revamped the process of making libraries. We've made it as efficient as can be. We've drastically miniaturized, allowing us to move faster, and we've made automation the basis of everything we do in the lab. One of the things I hope to talk with all of you about in the discussion session is some of the techniques we use to validate our DNA encoded libraries. I think this industry heavily focuses on the chemistry side, but I want to impress upon you that it's equally as important to validate on the encoding side of your molecule. And why is that? Well, the first thing is you should have the best DNA construct possible because your detector is PCR and NGS. If your DNA template is not a great template for PCR and NGS, you're automatically starting off with a blurry image. So I encourage all of you to make your DNA construct be the best it can be and to work with your PCR and NGS platform. In addition to designing a great construct, I encourage everybody to validate your encoding elements. We validate everything on the chemistry side, we should validate everything on the encoding side. And we can discuss afterwards, if you'd like, some techniques that we use in Anagenex to make sure our encodings are proceeding the way we desire. One thing I'd like to say is that we've focused on biosearch technologies as our preferred provider of oligonucleotides. And the reason we've done this is because we found biosearch to consistently meet the high quality demands we have on our encoding elements and also meet the time frame we need in order to make our libraries quickly. Now, on the chemistry side, I'm going to encourage you to also validate everything you do. And I'd love to discuss with you how we go about this as well at Anagenex. But in addition, I'm going to encourage you to leave no stone unturned with your chemistry. I'm going to encourage you to search by far and near for as many building block providers as possible. And I'm going to encourage you to include as many diverse building blocks as possible in your libraries. I'm also going to encourage you to really work on really interesting, really diversity forming new reactions, because after all, it is the chemical side of the molecule that is going to provide us with the drugs that we want to discover. So does it work? I'm going to present you with 2 case studies that we've used our platform to help us identify really interesting molecules for targets. The first is a challenging DNA binding protein target. DNA encoded libraries uses molecules encoded by a very large piece of DNA. If your target binds DNA, that obviously presents challenges in how you're able to use your libraries against that target. The second challenge this target provides is that it's a protein-protein interaction target. As we know, finding small molecule modulators of protein-protein interaction targets can often be difficult. The protocol we used to find modulators of this target were to select 500 million compounds in 10 different experiments and use our model to predict commercially available hits against this screening data. We also used our model to design a evolved library of 175,000 compounds, which could then be used again in that model to update and better predict compounds. So steps 3 and 4 were performed, used to update the model. The model then predicted more compounds to pick, and these compounds performed quite well. So our first predict, we did 500 million compounds, 10 conditions, and we screened these results against 20 million commercially available compounds. From our ranking, we picked 135 compounds to test. Of these 135 compounds from our prediction 1, or what we call ML1, 21% of these compounds confirmed engagement with our target. More importantly, 2/3 of these compounds showed desired disruption of the protein-protein interaction. Over here, you're seeing the spider plot of the chemical properties of the ML predicted compounds. You want to be inside the red dash, and as you can see, our molecules are of good chemical quality. But most importantly, down below, we're comparing our compounds to the best known literature standard, which is this compound here, which was tested in the clinic. And you can see that we have many compounds that are equal to or significantly better than this compound from our first round of predictions. But don't forget, we also designed a library that's going to help a second round of predictions. So let's see how that looked. So when we designed our library, rescreened it, and then used that data in our machine learning model, our predictions went from 21% off the initial data to 58% on the new data. And we now have many, many potent molecules with good chemical properties against this target. My second case study is a synthetic lethal oncology target. The goal of this program is to find selectivity of one target against a very close family member of it. So we're looking for selectivity of synthetic lethal 1 over selectivity of synthetic lethal 2. The protocol we did here is a bit different. The first thing we did was we screened more compounds, and we used those compounds to train and predict from commercially available catalogs, just as we have done in the previous experiment. But the library we designed here is actually quite different. This evolved library is used directly in biochemical and cellular assays to predict true activity. So in the previous case study that I gave, our prediction from the evolved library was on biophysical or binding. This library was designed to give biochemical and cellular, so true activity measurements. We then use steps 3 and 4 to update our model and do a second round of predictions. Now, this story is incomplete. We are currently at step 6, but I would love to show you our progress through step 5. So here's our initial selection. We did selections in a way that we could determine selectivity. So on this axis, I'm showing molecules from our DELs that were selective against target 1. On this axis, I am showing molecules from our DEL that were selective against target 2. And in the middle, I'm showing you molecules that are hitting both targets. This data from this selection was used in our model to predict commercially available compounds, or what we call ML1 compounds. And as you can see by this plot over here, we found a very nice chemical series that was 15-fold selective for one target over the other. As I mentioned, we also designed a library that allowed us to measure activity directly, and this library was screened in a biochemical assay under single point conditions, and we found many potent hits against both targets in single point analysis. We then carried these potent single point hits into dose response, confirming very active, very potent, and most importantly, selective molecules to our target. Some of these molecules have been tested in cells, and these experiments also show potent activity of our compounds against the desired target in cells. As I mentioned, ML2 is in progress. We are very excited about this experiment, and we are expecting to have some really good compounds come from this experiment, which we will quickly move to cellular and eventually animal studies. So, in conclusion, Anagenex is pretty new. We've only been around for about 2 years. In 2 years, we've built a platform that has identified 10 machine learning identified hit programs. So machine learning hits against 10 different targets. We execute our platform in 4 to 6 months through 2 iterative cycles. And we currently have 4 active programs which we are pursuing clinical development with. Thank you for your time today, and I look forward to discussion with you after this presentation.
Thank you, Joe, for that interesting presentation. I hope that everyone listening enjoyed the webinar and found it informative. We'll now move on to the last part of today's webinar, the question and answer session. Just as a reminder, before we get started, please feel free to continue asking your questions using the tab at the left of your screen. Simply hover over the purple speech bubble icon and click to ask your question. So the first question I have for you, Joe, today is, what encoding method does Anagenex use? And could you please describe what DEL is in more detail?
Sure. So first, I'll take the second question first. So DNA encoded libraries is essentially a screening technique. So many people are familiar with high throughput screening or fragment or structure-based design. DNA encoded libraries is just another screening technique. And so how the technique works is you use a DNA barcode to encode what the compound is on the other side. And this DNA barcode essentially allows you to mix, billions and billions of compounds together in the same tube and screen a condition in that tube against these billions of compounds, because at the end, all you have to do is put this barcode into a DNA sequencer, and you can read it out and tell which molecules are hits and which aren't. So we're not confined to 1536 well plates with a single compound in each well. We can use one tube as equivalent to a well, but screen billions of compounds. We practice what's called DNA recorded synthesis. So there are many different kinds of DEL. The kind of DEL that we do, and which I would say is by far the most practiced, is DNA recorded synthesis. And that simply means we have a DNA barcode on one side, some sort of linkage in the middle, a small molecule warhead on the other side. And again, we use PCR and NGS to read the sequence of the hits and tell us what the molecules are.
Right, thank you. Lots of questions coming in. The next one asks, could you please elaborate on some of the techniques that you use to validate your DNA tags?
Sure. So I'll give you just a few. We actually do quite a bit, but the first thing we do is we don't assume that anything that comes through the door is actually what we're told it is. So everything every oligo that comes through our door gets tested in a ligation reaction, and we run gels on every single tag to make sure that it will add to and be added to, so it's complementary both ways. We also check all tags by LCMS to make sure that they add up to the right mass. We make DNA-only libraries, so where we do nothing on the chemical side of the DNA, and we put those libraries into dilute library sequencing to make sure that the tags are represented in the proper distribution. And we also do dilute library sequencing on every library, because how you purify a library will impact its tag distribution. So the DNA-only library will give you the distribution of how the tags perform when you're not doing any chemistry and potentially removing unchemistried DNA. But the DLS in libraries is more to tell you your tag representation based on the chemistries and purifications you did.
Right. No, thank you. The next question that's just come through asks, what is the final library purity and yield using the LGC Biosearch Technologies DNA? And the second part, which T4 DNA ligase do you use?
So second one, much easier to answer than the first. So we use Biosearch's ligase. We've again evaluated many sources. There's lots of ligase that you can buy. Some companies even make it themselves, but we use Biosearch's, and we found it to be very good. And so the final purity of each library, it's a tough one to answer, because so if your tags are good and you don't remove any unchemistry, then the final purity of your library should be 100% the full-length DNA you want, and that's that. But if your purification strategy is to try to remove incomplete chemistry, then your final DNA balance is going to be different tag by tag based on the chemistry you did. So it's a little bit difficult to answer that, but I think what I would say is our final libraries are of very high quality, and we understand that DNA encoded libraries is a combinatorial technique, and so combinatorial techniques don't make 100% what you want all the time, and we've put a lot of time into understanding when something doesn't go to 100%, what it leaves you with, and how to interpret that data.
Right, yeah, thank you. Lots of questions to get through. The next audience member asks, what is the average tags purity that you expect from the vendors that you use?
Oh, very high. It has to be really high, or else this technique won't work. And so, much greater than 95%. And I can say, validating thousands and thousands of tags over my career, we've maybe failed definitely less than I have fingers and toes, but probably less than I have fingers, over tens of thousands of oligos. Very few fail.
Right, yeah. The next question, how many million reads are necessary to sequence a library and obtain a useful data set to apply to machine learning?
Okay, so I'm going to approach that question a little differently. So we select libraries in pools at the very first stage. So we have 2 billion right now. We have 2 billion. It's always changing. Molecules that go in. We count the number of molecules from each library. So every molecule from every library goes in at the same number. And our goal is to sequence 100% of the selection output. So we believe in a lot of molecules in. We believe in selecting a high selection coverage at the end. We aim for 100%. And that's what we believe gives us a clear picture for us to do our analysis on. If you do one of those things, a lot in, or deep sequencing, it's going to help improve the image. But if you do them together, it's going to empower your analysis.
Right. No, thank you for answering there. I have a follow-up question asking, how large does a library need to be to generate a large enough data set to train your proprietary machine learning models?
So our largest libraries, again, remember, we do these things in pools. So, right now our library that's going in is 2 billion, even though it's 30 different libraries. But our library size range from 100K ish to a little over 100 million, and so that's kind of the ballpark we play in. 100K to 100 million, and then combined together.
Mm-hmm. Right. No, thank you. The next audience member has submitted a question asking, what are the limitations of DEL, if any?
I mean, to give a very business development type answer, I would say the limitations of DEL are your imagination. I mean, you see a lot of people criticizing the types of molecules DEL make. They're not the types that medicinal chemists would want, or the types of proteins that DEL can select, which, a lot of people focus on solubilized proteins, and those just simply aren't true. I mean, we've done analysis of published medchem molecules, and can we make them in DEL or not? And we can make greater than 85% of what med chemists are publishing. So, and then, you see lots and lots of papers coming out about people selecting, all kinds of different proteins that aren't just solubilized. So I like to say, what do you want to do with DEL? And if you know what that is, go figure out how to do it. And that's why I say it's really only up to your imagination, because you need to figure out what you want to do and then how you want to apply DEL to it. And, I mean, I've been challenged over my career to develop reactions that, people would say are impossible in DEL world. And we've done it many times over. So just, if it's important to you, figure out how to do it.
No, thank you. Different question come in asking, how often have you observed a difference in the behavior of small molecules when on DNA versus off DNA?
So I'm going to answer that like if you selected a molecule, assayed it on or off DNA, typically what we see is things like potency increase on the molecules off DNA, but that's not 100%. But I think maybe to take this question to one of the criticisms of DEL that I tried to answer in the last question, a lot of people will report, they do a screen and then they follow up the molecules, and 50% of what they follow up with off DNA chemistry is active and 50% is not. And so I can try to answer this question from that regards as well. And I would say that's all about how you design your platform, and that's all about the questions you ask in your selection. And so if you design a robust platform where the analysis is good and you design selections in a way that allow you to answer a question, is this molecule going to engage in this target this way or that way? Then what we find with our platform is when we make compounds, we have a very low rate that don't confirm. So when we do our analysis of a selection and we pick compounds to make, those compounds confirm, because again, we're doing selections in a way that are looking for a specific mode of action. So therefore we can test it. What I see a lot of people do is very limited conditions and then do a selection, and then they put it into a biochemical assay, which is completely different than any condition they tested, and then wonder, why is it my molecules didn't work? It's like, well, you didn't test them the right way. And then another thing I see commonly happen that leads to a low confirmation rate is people don't understand what they've made. And so, what they've selected is something different, but happened in chemistry than the molecule they're making. And so we've put a lot of effort into validating building blocks and understanding what happens when a molecule doesn't go to completion. And we put a lot of effort into understanding how to analyze this so that we can not only use it to make the right compound from the DEL, but that we can use it in machine learning so that machine learning knows the right compounds to focus on, right? Like that's really dangerous in machine learning. If you're telling it this is a hit, but it's not actually that structure, then machine learning is going to be tricked. So you need to really understand the stuff you're making.
Yeah, no, right. So it's quality inputs there as to how it's taken forward. The next question sort of focused on Anagenex, asking why you why you focus at smaller libraries than other most Dell shops are reporting.
Sure. So that gets back to one of the things I was talking about with one of the earlier questions. We believe in balancing our inputs. So every library molecule from a different design gets put in at the same number of copies. And we believe in deep sequencing on the output. And we believe what that gets you at the end is a much clearer picture of the questions you've asked, and therefore it helps your analysis. So, you can do that with larger libraries, but it gets challenging. And so I think what you start losing control of with your larger libraries is the noise, right? So the process starts becoming a bit noisier, and you just don't get as clear of a picture.
Right. The next question that's just been submitted simply asks, how do you go about purifying your DEL?
Sure. So, reverse phase HPLC. We've sort of customized our gradients library to library. And so, we put a lot of effort into, getting the most out, of what we want. But yeah, it's all reverse phased HPLC. And yeah.
Yeah. No, great. Thank you. The same attendee asks, DEL data is inherently noisy, but what kind of techniques do you use to eliminate the noise to avoid feeding the machine learning model false positives?
Sure. So I think the most important thing is validating your building blocks and your tags, and validating your reactions, right? So making sure that things are going the way they should be when you move into a library. We really focus on, so as I mentioned earlier, DEL is a combinatorial technique, and it doesn't produce, every well that you do one thing in, it doesn't produce just one thing, right? Nothing ever goes 100% to completion, or I shouldn't say never, very rarely goes 100% to completion. So when it doesn't, make sure you understand what the other stuff that is generated, because the DNA tag tells you the recipe for the mixture. It doesn't, most people look at it as it tells you this structure. No, it tells you the recipe you did and a whole bunch of other structures that you made. And so we really focus on understanding that mixture, and we really focus on being able to tell machine learning which part of that mixture is what's causing the event against your target. And just to clarify that a little bit more, if you made a 3-cycle library, so you put 3 pieces together, just focusing on the chemistry side, let's assume the encoding side goes 100% every time. If your building blocks at each of those 3 steps were 70% product, 70% product, 70% product, the actual structure you intended to make is less than 35%. So that means 65% are other structures. PCR is your detector. PCR is super sensitive, way more than any biochemical technique. So therefore, if it's one of those other, peaks that makes up that other 65%, there's a good chance you're selecting that. And so we put a lot of effort into understanding which of that mixture is being selected.
No, thank you. Thank you for kind of describing your process there. I'm sure, they found it really useful. A few more questions coming in. The next one asks, when you follow up with an evolved library for biochemical screening, are those compounds still synthesized on DNA?
So we follow up a whole bunch of ways. And so when, if it's just simply do a DEL selection and then follow up the compounds, we have a bunch of techniques. So, we'll measure things on DNA by SPR. We'll make compounds on DNA with a cleavable linker, then we'll cleave the compounds and test them as true small molecules removed from the DNA. And we'll also do what I call powder in a vial, where we just look at a selection. We say, these are the compounds I want to make from that selection. We'll make a MIG or 2 of them as a highly purified small molecule and test it that way. But we follow up compounds a lot of different ways. And then don't forget, we also follow up our selections with machine learning designed libraries to also test. So lots of ways to follow up compounds, and some on DNA, some off DNA. Right. But remember, the drug is off DNA, so eventually you got to make some powder in a vial.
No, thank you. We've got time for a few more questions. The next one sort of in a couple of parts. The first being, when you say that machine learning informs selection of commercially available compounds, do you mean building blocks to incorporate into subsequent DEL runs? And the second part, which building block vendors would you recommend?
Okay, so the first one, so we do two things, and we do them multiple times. So the first will be to predict screening compounds from commercially available catalogs so they're true small molecule drugs. They're like 350, 400 molecular weight, have the properties you want, and machine learning will look at the catalog of available compounds and say, based on the DEL screening data, these are hits that you didn't test that if you bought these, they should be hits. And so a couple of things I want to point out there. These compounds look nothing like the DEL compounds, as I showed in that UMAP. So we're not using machine learning to predict commercially available compounds to buy that look like our DEL compounds, because we just make the DEL compounds, right? We already know what they are. We're using machine learning to help us advance chemical space, but predict hits of molecules that look completely different. So, and those are similar to those are compounds you might buy to build an HTS screening deck, right? Like people who have 2 million compound HTS screening decks, they have to go somewhere, and they go to these commercially available catalogs to help build out their deck. The second thing we do with machine learning, and we can do 1 and 2 as many times as we want. The second thing we do with machine learning is have machine learning design a library that, if we had it, would have been chock full of hits. So we give it parameters, right? We say, like, this is what's feasible, and then we give it building block parameters and say, this is what's available, and the machine will say, based on what I'm seeing from the initial DEL screen, if you build this library, it's going to help me laser focus in and make better predictions because this library is going to be full of high quality hits. So we do those two things. The second question, I mean, I would say get busy with Google. Figure out how to find. I specifically use that clip art of a person looking under a rock for a reason. Like, leave no rock unturned. Find really great chemical vendors, and so. I talk to a lot of people and all they do is search enamine. I love enamine. I love the people at enamine. We spend a lot of money with enamine, but there's a lot of other really great vendors. And so actually just published today on LinkedIn is a collaboration we have with Liverpool Chirochem, which is in the UK. They're what I would call a niche vendor. So they produce really high quality, high end, so read expensive building blocks, and very different from the building blocks you're going to find at enamine or Aldrich Market Select. And we have a partnership with them, and we're actually doing validations on their building blocks and allowing them to share that data with companies who may be interested in purchasing their building blocks. So we've made it a lot easier for people. If you want to make a library that includes those building blocks, they can share their catalogs and you can see how they validated under Anagenex conditions. And make purchases from that if you want. But that's just one example. There's lots of other really cool sources of building blocks. Sometimes academic groups, making stuff, and you can tap that and make some yourself. Embrace automation and figure out how to make really cool building blocks yourself.
No, brilliant. Thank you for your top tips there. Final question that we have time for today, and thank you, Joe, for your time. Please, could you elaborate on some of the techniques that Anagenex uses to validate those building blocks?
Yeah, sure. So, we feel validation is key to not only making high quality libraries, which will help machine learning, but just using those results as well in machine learning to help understand what the key molecule from that mixture is. And so I think we have a pretty exhaustive way to validate building blocks. So one of the things we do is a building block doesn't just get validated on one substrate. We validate a building block against multiple different substrates. And so think about it as sort of an easy to pass hurdle, a medium hurdle, and a difficult hurdle. And we'll validate our building blocks against all of those and see how they perform. We also, if building blocks have multiple handles, so meaning they go on to something and then accept something else, we validate both of those handles in the way I described. And so, we want to make sure that things not only attach well, but that they can do the next reaction quite well. And we've even done some pretty cool stuff where we'll save retains of previous cycles of libraries and we'll validate new building blocks against those. So, there's thousands of things in a well. We're not going to ligate it. We're going to sort of leave the mass of the DNA the way it was and just see how well do our building blocks do against thousands of things. Now, the analysis of that is a little bit of an art because you're not going to tell if, you can tell it reacted with 70 or 80 or 90% of the pool or less, but you're not going to be able to tell which one of the things in the pool it didn't react with. But in general, right, if it did really well against that library retain pool, and then it accepted well, then it's a good indicator that it's going to do well in whatever library you want to use it in. And so it's a good way to evaluate that. And then the key is record all this information in a way that's usable in your analysis. And so make sure what molecules you want to make at the end, right? because it's not always the three things put together that you think it is, so, or you want it to be. So cool.
Yeah, brilliant. I'm afraid that is all that we have time for today. I'd like to thank our expert speaker, Joe Franklin, for today's discussion, presentation, and this Q&A session. And a big thank you to everyone joining us online for submitting your questions. We hope that you found this a really worthwhile webinar. If you have any other questions, of course, please feel free to email myself using editor@selectscience.net, and I'll follow up with these after the webinar. And as a reminder, you can download related resources in the tab at the left of your screen. This also includes a certificate of attendance. And if you'd like to listen again to today's webinar, or perhaps invite a friend to listen, it will be available to watch on demand in just a few days' time. Goodbye, and thank you once again for joining us.